All right, here's the an interesting application of stereochemistry. All right, mentioned before at the beginning that of this chapter that chirality is in everywhere in our body, in a cell, in our DNA, in a protein. It's everywhere. Okay, but we can use this. Give you some example. All right, let's take a look at the D-glucose. Right, this guy rotates planar polarized light clockwise and L-glucose counterclockwise, just enantiomer of the D-glucose. Okay, so let's say we eat sugar, right? We have glucose in our body. Glucose will be converted to ATP using a citric acid cycle or Krebs cycle. You probably have heard about it, right? And in order for glucose to get into the citric acid cycle or Krebs cycle or become ATP, D-glucose have to pass through one enzyme called hexokinase. Can you guess what does this mean? Hex means 6, right? This enzyme process 6-membered ring sugar. Yeah, we call it hexokinase. Okay, so and you guys probably have learned before in biology class or whatever that the substrate and the enzyme have to fit perfectly, like not a lock and key. We don't use lock and key anymore. We use glove and hand because it's flexible, right? It's not like lock and key that lock and key kind of imply that the object are rigid, like they cannot move. But enzyme molecules can move, right? Enzyme is a protein in solution. They can get larger, get smaller. They can move around. So we prefer to use gloves and hand. Okay, so you have to fit perfectly with glove and hand. All right, right there, fit perfectly. All the OH group, the structure, everything fit perfectly with the active site of hexokinase. And once it happens, it goes through the Krebs cycle glycolysis and then produce ATP. If you eat a lot more sugar, you get fat, right? Because produce more ATP, they go to our body have to take the glucose, extra glucose, and convert it to fat. Now, L-glucose, if you still remember, we talk about L-glucose as a sweetener. You can eat as much as you want, you never get fat. Have you ever wondered why? Here's the answer. L-glucose cannot enter glycolysis or Krebs cycle because it cannot pass through hexokinase. It doesn't fit. That's what she said. It doesn't fit. And so hexokinase cannot recognize the structure of the L-glucose. So what's going to happen to the L-glucose? It just pass through hexokinase, stay in your blood system, and eventually you're going to pee it out. Right? That's how sweetener works. If you eat a lot of sweeteners, you can test your pee and see if it's sweet or not. Please don't do that. No, don't do that. All right, some type of sweetener. We have stevia, 250 to 300 times sweeter than a regular glucose. We have saccharin, 300 to 400 times sweeter. Sucralose, that's hard to say, sucralose, up to 1,000 times sweeter than regular glucose. Okay, and look at the structure, really close, right? Sucrose can be processed by hexokinase, look like that. Hexokinase recognize it. Sucralose, hexokinase cannot recognize it because of what? Chlorine in there. Chlorine is really big, and that's a point down like OH in this OH either. This chlorine point up, not gonna fit into the hexokinase, the active site. Okay, and you eventually will pee this guy out. All right, chlorine is really big. All right, last topic for this chapter.